In December 2024, Moyang Liu from Shanghai Jiaotong University and Liang Heng from Hainan Academy of Agricultural Sciences published an article titled Polyploidy Drives Autophagy to Participate in Plant-Specific Functions online in iMeta. Autophagy is a highly coordinated biological process, and its core mechanism is the gradual formation of autophagic vesicles through autophagy coproteins, which encapsulate substrates and transport them to specific areas for degradation. This basic mechanism is highly conserved in different eukaryotes, but with evolution, autophagy has gradually been involved in more biological processes. For example, most plant autophagy genes are difficult to complement the autophagy defects caused by knocking out homologous genes in yeast. At present, little is known about how autophagy develops specific functions in plants. This study used multiomics data and evolutionary methods to explore the evolutionary mechanisms that drive plant-specific autophagy functions. This study used nearly 10,000 data samples from public databases such as GEO, ArrayExpress, and CNGBDB to identify 997 core autophagy cooperating genes in Arabidopsis at the transcription, transcriptional regulation, and protein interaction levels. Using the MMAD tool, a wide range of connections between autophagy and other biological functions were established, including 3,419 autophagy cooperating functions such as mRNA processing regulation, growth-negative regulation, and programmed cell death. In order to reveal the evolutionary driving force of plant autophagy synergistic function, using the above 997 genes, 92,967 homologs of core autophagy synergistic genes were identified in 77 plant species. The proportion of these genes in land plant genomes increased significantly, accounting for 1.19% to 7.47% in representative genomes. In terrestrial plants, polyploidization is the main evolutionary driving mechanism, and polyploidization promotes the formation of autophagy synergistic genes in terrestrial plants. Overlap analysis found that humans, yeast, and plants share 50 common autophagy functions, including cellular responses to unfolded proteins, lipid oxidation, and modification-dependent protein degradation, but yeast does not have unique autophagy functions. Plant-specific autophagy cofunctions include chloroplast division, root hair elongation, and cell wall macromolecule decomposition. In monocots and dicots, autophagy co-genes were found to have gene loss and redundancy during polyploidization, resulting in changes in co-functions. Biological processes related to chloroplasts, such as regulation of photosystem II, chloroplast division, and chloroplast starch granules, are unique functions of photosynthetic plants. MMAD analysis showed that autophagy is related to multiple chloroplast functions, including chloroplast division. Authors' previous study, 5,274 chloroplast division cooperating genes, were extensively annotated. Overlap analysis of chloroplast division cooperating genes and autophagy cooperating genes showed that the two shared 4,258 genes, including 179 high-confidence genes, known chloroplast division genes ARC6, MIND, YLMG1-1 and the autophagy gene ATG8B. It was further found that these chloroplast division genes were significantly expressed in the autophagy state. Based on multiomics data of nearly 10,000 samples from 77 species including Camellia oleifera, tomato, and Arabidopsis thaliana, this study established a method for plant functional evolution, systematically evaluated the evolutionary laws of plant autophagy functional diversification, involved in biological processes such as chloroplast division and flowering, emphasized the important role of polyploidy in the adaptive evolution of plants, provided a new perspective for plant autophagy research, and provided important theoretical support for plant breeding and functional genomics research.